These are notes on the structure of DNA, and it's found on pages 513 to 518 in your book. We'll also cover DNA replication that we talked about in the cell cycle when we talked about the S phase of interphase. So we'll look at that as well. So a quick flashback to macromolecules, because if you remember, we actually talked a little bit about DNA when we talked about macromolecules, and then I said, but we're not going to worry about that right now, we'll come back to that in a couple months. Well, here we are back to that. So if you remember, in our body we have 4 basic polymers or macromolecules. We have polysaccharides, and their monomers are monosaccharides. So like we have starches, and those are made up of glucose. We have proteins, and their monomers are amino acids. We have lipids, and their monomers are the glycerol molecule and then fatty acids. And then we have nucleic acids, and their monomers are what we call nucleotides. And so DNA at its heart is a molecule. It's a molecule that we have in our bodies, in our cells. We know it's in the nucleus. And what's interesting about DNA is a lot of us understand sort of intrinsically that it's the molecule that makes us us, that codes for everything our body does and is. But what's really cool is that in every cell in our body, with the exception of our sex cells, there is the same DNA. So I have the same DNA in my toe as in my heart as in my brain cells, and yet it knows to make the cells do different things in those different places. So that's kind of cool. But that's why if you go leaving your DNA around a crime scene, it doesn't matter if it's from your hair or from the inside of your cheek or some saliva or some skin cells, it's all going to be the same DNA, and it'll all be traced back to you. So DNA is arranged into chromosomes, which are built from the nucleotide monomers. And the chromosomes get packed together, like so, to take up less space. And I don't remember if I talk about it here or if I'm planning on talking about it Friday when we do a lab on this. But the chromosomes get wrapped around a bundle of proteins called histones. And that's what helps them get packed together to take up less space. Because if you're to unravel your DNA, it's like miles and miles of DNA. And yet it all has to fit inside your cells. And so a chromosome is a single molecule of DNA, but this whole thing is one molecule. It's not like each part is a little molecule. It is one large macromolecule. One might be able to argue that it's the largest macromolecule in your body. So, as I said earlier, DNA is made up of monomers called nucleotides. And the nucleotides have 3 distinct portions. They have a phosphate group, and so this is the phosphate group. And remember that phosphate, as we've been talking about all along with our phospholipids, that carries a negative charge. Can't really see that, so I'm going to put that there. That carries a negative charge. A deoxyribose sugar. So hopefully you're recognizing this ribose. The ose means it's a sugar, so it's a carbon, hydrogen, and oxygen compound. And for those of you who want a little more specificity, this is actually an oxygen right here. And then there's 5 carbons. There's a carbon here, there's one there, there's one there, there's one there, and there's actually one there. And then what we call a base. And it's the base that makes different nucleotides different. So every nucleotide is going to have the same phosphate group and the same sugar group. But then the base is what makes the nucleotide unique. And there are 4 bases. So the 4 bases of DNA are adenine, guanine, thymine, and cytosine. And one of the things to notice is that adenine and guanine both have, wait, I've lost my mouse here. I don't know where it is. Anyway, adenine and guanine both have, oh, there it is, both have 2 carbon rings, whereas thymine and cytosine only have one carbon ring each. And so that's going to help us when we talk about the structure of DNA, because if you think of DNA, think of it as like a ladder that's then a twisted ladder. But each rung is approximately the same width. Well, for that to happen, that means you can't have adenine bonding onto guanine, because that would make like a larger rung than thymine bonding onto cytosine. So this is going to drive how some of these things bond together. The other thing I want to point out is in each of the bases, there are a lot of nitrogens. And so often you'll read that the bases are nitrogenous bases. That means containing nitrogen. And you'll see in each one, there's a significant amount of nitrogen. And we abbreviate these bases by their first letter. So we'll just call this one A, we call this one G, we call this one T, and we call this one C. And so get used to those four letters, A, G, T, and C. Later we'll actually bring in another one called uracil, but we're not going to deal with that until we deal with RNA. So back to this idea of the nucleotide. Here you can see this, I believe, is, let's see, adenine. Yep. So this is adenine. And you can see what it looks like with, instead of shapes, the actual bonding of the molecule. So here's our phosphate group. These negatives here are why the phosphate group is negative. Here's our sugar. There's that oxygen I was talking about earlier. And then here's our, and right here you see it, nitrogenous base. And this one is the one for adenine. And then each one, if you go back to here, would be bonded on to the same carbon. So they're always bonded on to this carbon right here. So, as I said before, nucleotides always pair specifically because otherwise these rungs here, so if you think of the ladder, this is kind of like a ladder on its side, or some people will think of it as a zipper. A zipper is actually not a bad analogy, probably better than a ladder. But either way, here are your kind of the supports of that zipper. And so in order to make it work, these have to always be the same width. And so therefore you can't have like a G pairing with an A, because those are larger bases. So in this, A always pairs with T. In this, I mean in DNA, A always pairs with T and G always pairs with C. And they bond using hydrogen bonding. So notice that between A and T there are 2 hydrogen bonds, and between G and C there are 3 hydrogen bonds. So if you go back to your knowledge of water and talking about hydrogen bonding, remember these aren't actual bonds, they're more forces of attraction between 2 molecules. We often call this our phosphate backbone, because the phosphate is kind of what's holding it all together. Sometimes you'll hear of it as a sugar phosphate backbone, because of course you have your ribose here. And then what you're going to hear a little of, and I don't want you to dwell too much on it if it's a little too confusing, is you're going to hear about the 5' end and the 3' end. And basically if you number the different carbons, so this would be our oxygen. This is carbon 1, this one is 2, this one is 3, this one is 4, and this one is 5. And it's going to be like that on every single one. So you'll notice the 3' end is the one where kind of the 3rd carbon, this one here, is at the end. And here the 5' end is leading by the 5th carbon. And what that means, though, as far as the structure of the DNA, is that if you think of 5' to 3' as maybe right side up, then 3' to 5' is upside down. And so these 2 strands are the reverse of each other. All right. So then you don't just have this kind of ladder structure, it's a twisted ladder, which forms what we call a double helix. And James Watson and Francis Crick definitely get the credit for discovering the double helix structure of DNA. If you're interested, you can look up like the history of how we figured out what DNA looked like, and there were all these different ideas and hypotheses, and then finally we realized, by we I mean Watson and Crick, sort of realized it was a double helix. Now, within science, though, there's a little bit of a controversy, because really it was this woman, Rosalind Franklin, who discovered that it was a double helix using X-rays to kind of tell what the structure was. But for a number of reasons, one of which being she was a woman at the time, not at the time, she was always a woman, she really didn't get the credit. And so a lot of people will say that Watson and Crick actually stole the idea from her. Some people say that they simply built off of her ideas. You can actually Google, there are like songs and parodies and even raps on YouTube about Franklin versus Watson and Crick, if you're interested in looking at some of those. They're kind of funny. So one of the things we talked a little bit about but didn't get into detail was DNA replication. And so remember we talked about during the S phase of interphase, the DNA replicates. Well, how does it do that? If you think about what it needs in order to replicate, first you would need to somehow, if this is a zipper, unzip these two sides of DNA. And it's always going to start unzipping at an area where there's a lot of A's and T's, because if you remember, A's and T's only have 2 hydrogen bonds between them, whereas G's and C's have 3 hydrogen bonds between them. So it's going to take a little less energy to zip apart the DNA at an A and T place. And I want you to think about if you've ever accidentally unzipped a zipper from the middle, like when your zipper breaks, that first unzipping is the hardest part, and then it all kind of comes apart easily. There's actually an enzyme that does that, and that's called helicase. So some of you may remember that usually, not always, but a lot of times enzymes end in ACE, and so that tells you it's an enzyme. And then think about, it's a double helix, so this is the enzyme that unzips the helix, helicase. Then what you need is you need extra kind of floating around bases to pair up with the DNA that was already there, or the half that's already there, and then to kind of fuse those two together. And that's done by an enzyme called polymerase. So again, it's an enzyme. There's our ACE, and then this is a polymer. And so it's an enzyme that makes a polymer. And some people, you'll actually see this written as DNA polymerase. So I'll put in the DNA there for you. So there we've got DNA polymerase. And then you have 2 identical DNA molecules. Half are the old section, that's the green, and half are the new section. And so that's how the DNA replicates itself to get ready for cell division. So this is actually as far as you guys need right now, but if you want more, so if you're kind of overloaded, you can stop the video now. If you want more, I'm going to show this video here, or it's more of an animation, and I'll talk through it, and that'll get into more details of replication. So if you think of this as being a really long strand of DNA, this side is going 5' to 3', this side is going 3' to 5', or 5' is on the bottom and 3' is on the top, so you can see that difference in orientation. And so first what we have is the helicase splitting the DNA molecule apart. And so you get almost like a bubble forming, and over here you can see that's going to happen at lots of different places along the DNA strand. And then this is talking about how helicase is always going to look for a section that's rich in A and T pairs. From what I was talking about, there's only 2 hydrogen bonds between A and T compared to 3 between G and C. And so it's easier to start this bubble, if you will, at a place that's rich in A and T. So then the next thing that happens is in the notes that I gave, I kind of skipped over this next part. So DNA polymerase is an enzyme, and as you learned earlier, enzymes have very specific functions. And polymerase, in fact, doesn't want to start replicating or doesn't want to start putting bases together until some of it has already been done for it. So kind of like you guys, how you don't always want to start your work until some of it's already been done for you. So there's actually another enzyme called RNA primase. You know it's an enzyme because it's got ACE, and think of prime like a primer, something that starts it. If you've ever like painted a room, often you'll paint with primer first. And so this is some RNA primase, and it's going to actually start the replication at some sort of initiation point, and we'll talk about initiation points a little later. One thing to note is that DNA always replicates from 5' to 3', so this side is going to be replicating down this way, whereas this side is going to be replicating up this way. And as you can see again in our long strand of DNA, that's happening in several different places. And so now you can see a couple things coming in. So DNA polymerase is starting to bind base pairs or nucleotides up here, and again it's going from 3', or it's going in the direction of 5' to 3', so it's going down this way, whereas this one is going up from 5' is down here to 3' up there. And then you can also see there's more RNA primase up here because it's going to be happening in different places at once. All right. So you can see it's building lots and lots of fragments of DNA at one time, kind of filling in gaps. You can see over here, you're getting 2 strands of DNA. These sections of DNA that are divided by RNA primase, those are called Okazaki fragments, okay? But you'll notice you've got all of these fragments of DNA that have been built in between all of this RNA primase. But what you've really built is an entire strand of DNA. Now, the problem is we now need to get rid of the RNA primase because that's not part of the DNA. And some of the base pairs in RNA are a little different, and we're going to talk about that later. So then you have another enzyme, you can tell it's called ACE. This is called exonuclease, and that strips away the RNA primer, and then DNA polymerase comes in and replaces the primase with DNA nucleotides. And so that's how you get your complete section of DNA. And then you have yet another enzyme called ligase, which inserts any phosphates into any remaining gap. So if you look back here, there we go. You'll notice like right here, there's a gap where there's no phosphate. Here there's a gap where there's no phosphate. And so then the ligase comes in and inserts the phosphates. So in fact, there's a whole set of enzymes that work together in order to replicate the DNA. And then, as it says here, this DNA replication precedes cell division in mitosis, which we've already talked about, and meiosis, which we'll talk about in a couple weeks. So that gives you a little bit of a more detailed showing of what happens during DNA replication. If this was too much, all I really want you to know is what the helicase does and what the polymerase does. Please put any questions you have on the comment section of YouTube or ask them in class.